Hi, everyone. My name is Jaqueline Picache, and I'm a postdoc at NCATS. Today, we'll be discussing with you my development of a novel label-free, high-throughput drug repurposing screen. When developing a high-throughput drug repurposing screen, we have a couple of considerations, and those include speed. We want our assay to be as fast as possible so we can analyze as many samples as possible. Sensitivity, we want lower limits of detection and quantitation to be as low as possible, and label-free. Typical screens rely on antibodies or fluorescent tags, which can make the drug repurposing screen pretty expensive when you're trying to do it in high throughput. So we are trying to do this in a label-free manner. I'm applying this drug repurposing screen to Tay-Sachs disease. And the main analyte of interest in Tay-Sachs disease is GM2, or ganglioside monosilate 2. After we develop this screen, we want to evaluate a library of drug compounds. And so keeping these two project goals in mind, our question becomes, how can we achieve these goals in a high throughput manner? I've developed a workflow here in order to answer this question. First, we take human fibroblasts and induce them into pluripotent stem cells. And from there, we induce them into neural stem cells. So Tay-Sachs disease is a neurodegenerative disease, and we want to analyze cells as close to the CNS as possible. Before we perform our drug screen, we wanted to make sure that our phenotype was showing up, and the phenotype being an increase in GM2 in these Tay-Sachs disease cell lines. And so we did that using fluorescent microscopy. So the GM2 is shown here in pink, and our DAPI is shown in blue. So we see that we have our phenotype and we can proceed with our screen. I plan to grow these cells in a high throughput 384 well format, and those cells will get dosed with different compounds. And from there, we will analyze them with multi-mass spectrometry. Our data output will look like this, where we have a bunch of mass spectra, and we will be analyzing our intact GM2 as well as the GM2 first fragment. Any hits that we find that reduce the GM2 within our Tay-Sachs disease cell lines will undergo validation and further development. Thank you for your time.